Hello everyone, my name is Mohd Waddah Almoufti, and today I'm going to be presenting to you my master's project. So my project is titled Investigating the Role of PGC-1α in Different Metastatic Breast Cancer Sites. So through my agenda, we're going to talk about some scientific background, which leads to my hypothesis. I'm going to show you some of the study design that I've been carrying out in the lab, and finally I'm going to be showing you some of my results. So scientific background, we're going to be discussing what is metastasis, what is PGC-1α, and we're going to be talking about a drug known as metformin. So my project revolves around stage 4 metastatic breast cancer. So the 5-year survival rate of a primary breast cancer, so when the tumor first is visible in the mammary gland, is 99%. However, once it enters that stage 4 metastatic breast cancer and that primary breast cancer starts moving to different parts of the body, that 5-year survival rate drops all the way down to 22%. So in my lab, we have a primary breast cancer cell line, or the control cell line, known as the 41. And we managed to actually, from the cell line, isolate 3 different cell lines. So using my laser pointer, we have the 590, the 2776, and the 533. And each one of those cell lines only metastasizes to its select target. So the 590 goes to the bone, the 2776 goes to the liver, and finally the lung, the 533 goes to the lung. And this is pretty much a big factor of why my project we're able to carry out my project in the lab. So what is PGC-1? So PGC-1 is a central regulator of metabolism. What that means, it represses some genes, it activates some genes. And it's highly involved with ATP and AMP and energy signaling. And my project pretty much is based upon 2 previous studies on PGC-1 that I'm going to be talking to you about. So the first one being a 2016 study by Professor Adele Walsh, where pretty much, as you can see here, I'm going to be focusing on the second figure. She found that in a high PGC-1 cell, those cells happen to be more sensitive to antifolate treatment. And in this case, they use the MTX, which is methotrexate. And why is that? That's because the increased PGC-1 through a drug known as metformin, and PGC-1 represses 1-carbon metabolism. So pretty much it stops DNA synthesis, pretty much stopping the cell from proliferating. So that pretty much worked as a synergistic effect with methotrexate and led those cells, the ones with higher PGC-1, to be way more sensitive to the treatments when compared to cells with lower PGC-1. So the second study we're going to be talking about is a 2017 study by Professor Andrzejewski, where she looked at those 3 sites I was talking about, the bone, the liver, and the lung, and she found that PGC-1 expression was not the same in all of them. So for example, the lung, shown in pink, was way higher, while the liver was way lower when compared to the 41s, while the bone was kind of similar. So this was pretty interesting, and this led to our hypothesis. Since PGC-1 is not the same in all the 3 sites, then methotrexate sensitivity should also not be the same. And that's our first hypothesis. If this happens to be true, we wanted to see, can we actually increase PGC-1 in those cell lines that are low? For example, in the liver here, can we actually increase this to go a bit up, leading those liver cell lines to be more sensitive to the methotrexate treatments, the antifolate treatments that we talked about in this study right here. So moving on, so that's pretty much our first hypothesis and our second hypothesis. And the way we're going to do that is by first quantifying how much PGC-1 we have before methotrexate and after methotrexate in all the cell lines. And we're going to do that through cell proliferation assays, so pretty much by counting every day, by qPCR analysis, and by Western blot. So that's our first aim in the lab. Our second aim is to do pretty much the exact same thing, but before we do any methotrexate treatment, we pre-treat with metformin. And metformin works by blocking complex 1 of the electron transport chain, which pretty much decreases ATP, increase AMPK, and AMPK will effectively increase PGC-1. And we want to see, would there be any difference in the way the cells react to methotrexate after we pre-treat with metformin. If that all goes well and good, the next thing we want to do is actually investigate the effect of all of this in a live model, in a mouse model. So those are our 3 aims, hopefully, in my 2 years in the lab. So quickly, I have a lot more results, but due to time, I'm just going to be showing you 2 graphs. The first one just being the control. So right here we have a proliferation assay, days 1 to 5. In this case, there was no methotrexate added. And as you can see, all 4 cell lines were approximately growing at the same rate. However, when we added 10 nanomolars of methotrexate, so day 1, there was no methotrexate added, and day 2, 3, 4, and 5, we added methotrexate, 10 nanomolars each day, and we counted each day. So as you can see here, the bone and the control were actually very similar, which is what we thought would happen since the PGC-1 levels are the same. However, the 2776 and the 533, so pretty much the lung and the liver, were way different. They reacted way different to the treatment than what we expected. So this was pretty much our first big finding, and what led us to go on and do qPCR and more analysis. But due to time, this is where I'm going to end my project, and hopefully I can continue showing you how we progress in the next few years. Anyways, thank you for listening to me. Don't forget to stay safe and social distance, and I hope everyone is doing well. Thank you.